Good afternoon, everyone. My name is Mugdha Lele, and on behalf of the TechX.in team at Venture Center, I'm happy to host the fourth wave of our Only One Theme Awareness Talk series on innovation management. As the organizer for this series, let me briefly first introduce the Venture Center and the TechX.in vertical at Venture Center, since this is the first talk of this fourth wave. As you all may be aware, Venture Center is India's award-winning leading technology business incubator for science and technology startups, where we specialize in supporting deep tech innovations by helping innovators and startups to commercialize their ideas. IP, technology transfer, and broadly technology management are very important aspects for any deep tech innovation. Venture Center has a dedicated TechX.in team which supports innovators and startups via various initiatives in the technology management domain. TechX.in is a technology transfer office supported under the Department of Biotechnologies, BIRAC, under the National Biopharma Mission. We offer several services and initiatives around technology commercialization via tech marketing, technology transfer, deal structuring, valuations and agreements, spin-off creation or new venture creations. Our team also offers support in terms of IP protection, search and analytics and policies, and support for institutions in innovation management. In addition to this, we are also actively involved in industry academia partnerships and contract managements. So feel free to reach out to us for any further details about these offerings. As part of all our initiatives, we also host several awareness workshops and training sessions. Yeah. We have already hosted 3 waves and over 50 plus talks under an awareness campaign on innovation management focused on one theme at a time, which is titled Only One Theme. And from today, we are initiating the 4th wave of this talk series. So welcome to the inaugural talk in this wave of Only One Theme. Today's topic is on patent landscape of CRISPR-Cas technology for medical applications. And our esteemed speaker for today is Dr. Malathi Lakshmikumaran. Let me briefly introduce Malathi ma'am before we begin the talk. Dr. Malathi has more than 30 years of experience in the field of biochemistry and molecular biology with an expertise in plant genomics, DNA fingerprinting, and genetic transformation. She serves as Executive Director and heads the Life Science Group at the IP division of the Lakshmikumaran and Sridharan attorneys. She is a registered patent agent herself and has been actively engaged in preparing, filing, and prosecuting of patent applications both in India and abroad. Dr. Malathi mainly works on pharmaceutical chemicals and biotechnology patent applications. She advises clients on plant variety protection and registration. She is also actively involved in the area of biodiversity and traditional knowledge. Malathi ma'am has successfully supervised several PhD students in the area of plant molecular biology and has more than 100 plus publications to her credit in various international and Indian journals. Malathi ma'am, with this brief introduction, I'm going to hand over the session to you. Welcome to the session.
Okay. Thank you. Can you hear me?
Yes, we can hear you, ma'am.
Okay, fine. I'm just going to share. Thanks for giving me an opportunity. I'm just trying to see how to share. Can you see?
Yes, ma'am.
Okay, I'm just going to make it full screen. Is that okay?
Yes.
Okay. Thanks, Venture Center and Mugdha, for giving me this opportunity to speak on a subject which is quite close to my heart. And it's a fantastic technology called CRISPR-Cas technology. But today, my talk will rest only with biomedical applications, though we know that CRISPR-Cas has applications in every field. It can be agriculture, it can be fermentation technology, it can be microbial, and it can be in any other technology. But today we are only going to talk about the biomedical applications. Though the technology as such will be very similar, but we will see how it is being used in the biomedical field. 1 minute. You can see my slide, no? No, I'm just going.
We can see.
Yeah, thank you. Yeah. So I will try to, do it for 45 minutes and then maybe have a question answer session. So I might rush through some of the slides. I'll try to spend more time on certain slides and not on all of them. What are the main things? I mean, CRISPR was well known, and CRISPR was a technology which was used by several bacteria against invading viruses because they don't have an immune system. And therefore, how do you now tackle any invasions by viruses that the bacteria will have to take care of? And so there is the three key phases are there's an adaptation phase, then there is a cRNA processing phase where the production of the CRISPR RNAs takes place from the CRISPR array, and then there is an interference phase where there is a detection and destruction of the invading nucleic acid. So as you see below what I've given like a line diagram, what is actually CRISPR is you have repeating units. These are called as short repeating sequences. And the color boxes I put blue, green, yellow are the viral sequences. So every time, and then we have the Cas gene. What people found that most of these bacteria, or all bacteria, have these repeats which go on and interspersed with some viral sequences, and they didn't really know. But once when they started studying it, they realized that these blue or the green or the yellow that I show are different sequences of the viruses which have affected the bacteria, and a certain portion, a small portion of the viral DNA gets integrated into the DNA and it resides there. So let's take an example that when I have a blue type of virus, which is, for sake of easiness, I'm saying blue, it attacks a resident copy is there. The next time that the same virus attacks the bacteria, it says, no, I know you. And a complementary RNA is produced, it goes and attaches itself. And finally, the Cas enzyme comes in and it breaks the viral DNA or the RNA. The reason is that if the virus uses the bacterial host to produce more copies of the virus, but if the viral DNA or RNA molecule is knocked out by the Cas because of the processing of the complementary RNA, or what we call as a CRISPR RNA, then you will not be able to have the full viral DNA, and therefore the virus is not there. So it was an extremely good immune system that the bacteria was using to defend itself against the attack of viruses. What this Actually, this system was known, but I think a couple of good scientists, one from the UCB that we call University of California at Berkeley, and the other one at Broad Institute, were working on this subject matter. But the first group which really came out and said, oh, I can use this CRISPR-Cas technology for gene editing was done by Emmanuel Charpentier and Jennifer Doudna. Jennifer Doudna is from University of California at Berkeley. Emmanuel Charpentier was at that time University of Vienna, and they both filed a patent application first before they published their paper in 2012. And once that paper was out in the open, people were working on it. Immediately, several companies, institutes, universities started working on it. And then came Broad Institute, which came up with the use of this technology in eukaryotic cells. So Emmanuel Charpentier and Jennifer Doudna showed that it can be used in any cell. But the experiments they did with the prokaryotic cells. What are the milestones? This discovery of unusual DNA repeats in bacteria was shown by the Japanese scientist Ishino in 1987. Also, University of Alicante had sequenced several bacteria having these repeat sequences, and they also identified this as part of the bacterial immune system. But in 2012, Doudna and Charpentier, they adapted this method for precise gene editing. And that has had great impact in all fields of technology, whether it's medicine, agriculture, or biotechnology. And there's been a surge in the patent filings and research over the past decade. Just to give you the timelines. So it was 2012, which is a very important year for us. And I might be coming back and forth for that year 2012, because that's a year in June 2012 that the publication happened. The patent application was filed by Jennifer Doudna and Emmanuel Charpentier, and it opened up this whole technology to everybody. And then we'll see how many other companies and institutes, universities started filing patents in this area. I've already talked about it that Ishino, University of Alicante were working on these, but they didn't realize the role that these CRISPR-Cas can play in gene editing. They did give the name CRISPR in 87, 2002, and identification of Cas9 to use to cleave the DNA was also known between 2008 and 2012. But in 2012 was the first time that they understood the mechanism of Cas9 cleavage, and in 2013 is when Broad Institute came and said, haha, I can use this technology in eukaryotic cell and we can actually treat a disease by this as well. And then, of course, a whole lot of things happened. You had the first clinical trial in 2016, and then the Nobel Prize was awarded to Emmanuel Charpentier and Jennifer Doudna in 2020. December 2020. And at that time, we also had Cas12, Cas13, which could be used for the detection of SARS-CoV-2 and other bacteria or viruses, which got a very quick approval and used it for detection methods. We'll talk about that as well. In 2022, multiple clinical trials are taking place. And therefore, one of the important things in 2012 was use of Cas9 cleavage to edit genes and so on. But if you look at it, we did have in Cas12a in 2015, Cas13 in 2016, 2017, which also will play a role, which I'll show you. And therefore, my talk today will focus on in medical applications, but in therapeutics, diagnostics, vaccines, and how CRISPR-Cas has also been used to come up with new cell lines, new animal models for drug screening, and the exploration of novel Cas enzyme for future medical applications. I'm not going to go too much, but you all should know that there are two types of class 1 and class 2. Two of the classes are there, but all the work that has been happened is on class 2, which is the bottom part of the figure that you can see. In this, you have the three important enzymes, Cas9, which is a class 2 and a type 2, which you'll see at the bottom part of my figure. And then you have Cas12, which is type 3, but again class 2. And then you have Cas13, which is again class 2 and type 6. So these are the three important enzymes that I will be talking about. Mostly in therapy, people have been using Cas9. In Cas12 and Cas13, a lot has been used in diagnostic, but people are planning to use and are using Cas12 in therapy as well. We will discuss about that as we go. In all this, the role of PAM is very important. What is the PAM? PAM is a sequence which is very close to where your complementary DNA, RNA binds. And without the PAM, the whole system of Cas nuclease recognition will not take place. And if the Cas nuclease recognition doesn't take place, then you will not have this breakage, either a double-stranded break or a single-stranded break, and so on. So it serves. So PAM is very important. And I will be talking about PAM because one of the patents which may get revoked is because they did not talk about PAM in their patent application. And so PAM actually serves as a signal for confirming the correct modification and what site that the modification has to take place. This I'm sure most of you all know, but I'll just do that. It is You do need a guide RNA. It has a tracer RNA. You can see in red the PAM. The complementary RNA goes and binds to the target. And when it does bind to the target, the whole thing gets triggered. The PAM is, of course, necessary. And once this is generally for Cas9, it is NGG. N can be anything. It has to have the sequence NGG, which has to be very close to your complementary sequence. And then you have the tracer RNA. The whole thing together we call as the guide RNA. So in the step 1, we have the guide RNA, which finds the target DNA in the cell. In the step 2, the Cas9 protein cuts the DNA strands. Generally, it can be in both strands. That was what first Cas9 came. But now a lot of modifications have happened. And you can also have single strand breaks, which are much better than having a double strand break. And then you have the replacement DNA, which then comes in, and you have the repair enzyme, which repairs this. So you can add in DNA, you can delete DNA, you can make base change, and so on. So let's look at the CRISPR technology. As I said, it's used a lot in research, in agriculture, medicine. We're only going to talk today about medical advancements that has happened in this technology. So, as I said, CRISPR-Cas is very versatile. It's cost-effective. It's a powerful gene therapy, and it accelerates in treating genetic diseases. The initial focus of Cas9 was only for genome editing, but now as we started seeing newer type of Cas, like Cas12, Cas13, you had new effectors, new type of PAM sequences, single strand breaks, double strand breaks versus that, and a whole lot of evolution based on Cas has taken place. This has actually expanded our scope, and we have got diverse health solutions which we can come. It also is very useful because it now can revolutionize disease treatments and in human health as well. Now, what did we do in this project? What we did in this project is to look at patent applications and patents. We went jurisdiction by jurisdiction, and we wanted to look at what are the geographical trends, which are the companies working on these, where are the innovation hotspots, and how people have worked on these things is what we were looking at. If you look at it in the global map that you see, you'll see the maximum number of patents or patent applications is in China, and it came out to be more than 10,000 patent applications. I'll talk applications, but they may cover patents, patent applications. They could be divisional, they could be continuation, and so on. There were 10,000 applications filed in China alone. The next came US with about 4,800 patent applications. And then, of course, you had Europe and Canada. Canada was about 1,800. Europe was about 1,700. Australia was about 1,600, and so on. India, we had about close to 1,000 patent applications that we detected or we identified in the Indian Patent Office. We don't have a robust way of searching in the Indian patent database. We've tried Derwent Innography, but we also had to use the Indian patent database. The other geographies are also Russia, but it's only 260. But if you look at Brazil and Argentina, are two sectors where we have seen a huge number of applications being filed. We tried not to put in some of the agricultural patents, but there is an overlap sometimes because some of the seminal patents would cover both agriculture and medical application. So you can make out very clearly that China, US are the major players in the field of CRISPR-Cas technology. There are other players, but they're much less in number as compared to these two giants. Now, we have something called as seminal patents. I will be talking about that in my next slide. But where are these real important patents or technology which was created or developed or invented are in the US. One is CVC, I call it a CVC, which is University of California, University of Vienna, and Emmanuel Charpentier. Now, Jennifer Doudna and Yannick worked in University of California. So the letter C is for University of California. Because Emmanuel was from Vienna, we have V for Vienna. But in many of the patent applications, especially the seminal, Emmanuel itself is an inventor and an applicant because she was moving places and so on. And she is also an applicant. So that is why we have CVC, which is for University of California, University of Vienna, and Emmanuel Charpentier. So whenever I say CVC, it is all three of them together. They hold several seminal patents and was a major contributor, and they actually changed the goalpost of how CRISPR-Cas technology can be used for gene editing. Of course, a Broad Institute comes next. They hold the foundational patents for CRISPR-Cas9 technology, but for eukaryotes. But if you look at CVC patents, it covers all, but we will talk about the patent interference proceedings at the USPTO. I'll just give a small, some details about that. So Broad was the first one in January 2013 that they filed a patent application and for eukaryotic use of CRISPR-Cas technology for eukaryotes, mainly humans, animals, and so on. Now, they also collaborated with MIT, Harvard College, and Rockefeller University. So some applications will be filed by all of them together. And the main person from Broad Institute is Feng Zhang, but not to mention that we also have a very good inventor who's a prolific inventor by the name of George Church from MIT. The Chinese applicants, they're highest in number, but they don't really go for seminal patent. But I will talk about Cas12, that they may have the patent for that enzyme. So Chinese not only having the highest number, there are some important patents that the Chinese applicants still hold. And very important for Cas12 for diagnostics that they do hold, and the company Shanghai Tolo, which we'll call as Tolu. So coming to this, everybody should know, people are talking a lot and saying, oh, will I be infringing on what patent, which patent I will lose, and so on. Out of these, let me just give you some information. And we need to know about these 5 different major players who are all holding the seminal patents or patent applications. And there is Vilnius University, again European. They were the first one to file the applications, and I've gone in that order. The University of California is what I call as CVC. We have Toolgen from South Korea. They have some very important patents on eukaryotic use of CRISPR-Cas technology for eukaryotes. And then we have Sigma Aldrich, now it's called Merck Millipore. And then your Broad Institute. So I've got 2 Broad Institutes. The first Broad Institute has MIT, Harvard, and Rockefeller. And the next one that Broad has filed is just MIT, Harvard, and Broad. So there is Broad and Rockefeller, and there's only Broad as well. There is a lot of fights that's happening between these seminal patents. All others which came are for application only. So let me give you a little insight into what is happening at the legal battle in the USPTO. So even though CVC was the first one to file the patents in 2012, that is May 2012, and in December 2012, Feng Zhang filed an application. But what Feng Zhang did from Broad Institute, he's a very prolific inventor and has lot of patents in his name in this area. He filed for expedited examination, which we call as a track 1 process, or the fast tracking of the patent to get a quick grant. Whereas CBC did not go because they were the first people to actually come up with this technology. And so they thought that they, being the initial or the first people to file such a patent, they would get access to the all technologies. but that was not what to be done. CVC was very upset that Feng Zhang went and got the patent granted in 2014, whereas the CVC patent of Doudna was not even granted by that time. And they were upset because they had filed for an application earlier, but did not get the grant. But Feng Zhang, being a smarter person, got this fast track application and got the grant. But his was for eukaryotic species, that is in animals, humans, and so on. They did go for an interference proceedings, what we call is CVC saying that the broad patent is application or the claims for it is same or similar to that of CVC, and therefore you cannot get a patent for somebody who's filed later and having the same invention. However, when they filed in 2012, it was the first to invent. So the question is, who was the first to invent the technology? There's no doubt that Jennifer Doudna and Charpentier were the first to invent. But the question then came, who was the first to invent this technology in eukaryote? And then came up the interference proceedings, which CVC filed against Braun. And the outcome in the US in 2017, earlier than that was the PTAB and then the US Patent Board of Appeal. They came and said, no, Broad was the first group to show the technology for CRISPR-Cas to be used in eukaryotic cell editing. And they said it's very distinct from that of UC's claim, that is University of California, which we call as CVC claim. They may be broad, but they did not have any experimental data or enablement for use of this technology in eukaryotes. All of their experiments were with prokaryotes. They may have had data, but they didn't go. And it was not very easy at that time. Whatever technology you did in prokaryotes, could you have also done it for eukaryotes? Can I use the same technology to use it and transfer this technology to eukaryote? So there's a question is yes, no, but definitely eukaryotic cell editing needed some more experimental work and some more changes to be done for it to be used for gene editing in eukaryotes. Therefore, Broad won in US, not that CVC patents was revoked. CVC patent was for prokaryote. Broad patent, which I call as Broad, MIT, and Harvard, were for eukaryotic cells. So now the question came is, if I go for a license, do I go to both? Do I go to one? Because by the time, because Jennifer Doudna and Emmanuel Charpentier were way ahead, a lot of the people went to them for licensing, but many people went to Broad as well. So now it becomes a dilemma, where do I go? Whom do I go for getting license for the technologies or patents that they have? Then comes the story in Europe. So you see, I've told you what happened in US. In Europe, Emmanuel Charpentier put a case against Broad Institute to show that they cannot use this technology in eukaryotes. And the reason they said was there was a little problem of the Broad Institute with respect to priority rights of their application. I'll just explain that about the Europe outcome. Though CVC won several of the major European patents for both CRISPR-Cas9 use in eukaryotes. Now in US, they said CVC has for prokaryotes, Broad has for eukaryotes, but when they reviewed it in Europe, they felt that a person skilled in the art, by knowing what CVC has done, can actually transfer the technology and actually do the technology in eukaryotes as well. So I said enablement is not an issue in Europe for CVC patents, and therefore CVC patents cover both prokaryotes and eukaryotes. Whereas in US, prokaryotes was for CVC and eukaryotes was for Broad. Then University of California did file a case. CVC did file a case against Broad saying you do not have the priority rights. What do I mean by that? The reason is when they filed the provisional applications, Broad, MIT, Harvard, and Rockefeller were the applicants. But when they filed the PCT application within a year, they only had Broad, MIT, and Harvard, and Rockefeller was out, including the inventor Marty Finney. So then CVC filed a case against Broad Institute saying that they don't have the priority rights. So they got the patent revoked of Broad, saying Broad does not have the priority rights of this application because all their provisional applications had Marie Finney as an inventor, Rockefeller as the applicant, but in Europe, they only had Broad, MIT, and Harvard, but did not have Rockefeller. And by the time they filed for this and the assignments, Marie Finney had not assigned his rights to Broad or MIT. All his rights were still with Rockefeller. So now Rockefeller had to give it to Broad, but when they filed the application, they did not do this. And so in Europe, whatever the applicants are there in the priority application, same should be at the PCT and in the national field. Whereas in US, you have a different system and you can change the applicants. So Europe, EPO did not allow that. And the seminal patent of Broad Institute, which they won in Europe, in US, was actually revoked in Europe only because of priority application, where Marie Finney as the inventor did not assign his rights to Broad. He assigned it to Rockefeller. Rockefeller did not assign it to Broad. Later on, they came and said, okay, now we have done it. They said, no, at the time of filing, Rockefeller should have been the applicant and Marie Finney should have been the inventor. Since you did not do that, your priority rights are gone. And so this is a complex licensing and there's a lot of ongoing litigation as well between Broad and CVC. The Toolgen versus Vertex. Toolgen is a South Korean company, and they worked on the ribonucleoprotein complex, and they used this for doing a gene editing in humans and animals. Now, Vertex were the first company to get the approval for use of the technology for sickle cell anemia and beta thalassemia to be used in humans. They got the approval in UK in November 23 and in EU in February 2024. And Toolgen then filed a patent infringement suit in UK against Vertex, saying that they are infringing on their patent and the patent number I've given. So don't worry about which. But Toolgen said, no, we have filed an invalidation case against the Toolgen patent because of enablement issues. And also they filed a nullity suit in Netherlands, arguing that their patent is too broad and they do not have all the experimental and enablement data to cover all the eukaryotic system. Toolgen said, no, we don't want to go and stop or block this regulatory process or the use of this technology for humans, but a fair licensing fees have to be given to them. And so this is not yet out. We should get the final court decision soon. The key areas we said is that new Cas enzymes, therapeutics, biological models, diagnostics, and vaccines. So as you know, we can use this PAM. I'm not going to go into this because most of this I've already done. But two important technologies that you should know is called as base editing, where I only change the base C to T or A to G, and including a DNA nick instead of a double strand break, which is called as a DSB or double strand break. These are just, you will only have a base modification. Then came prime editing, because all the cases cannot be just base editing. You might need to either delete a gene or you need to have modified gene inserted into it. And therefore, it came up the prime editing, where it goes beyond base editing, where I can have much wider modifications, including base edits, substitutions, insertions, deletions, without creating the double-stranded break. Now, it's very important when you get the double-stranded break and the repair mechanism takes in, you can have some issue. So there's only break in one of the DNA strand and not in the other, and therefore you have less problems of bystander effects or consecutive base changes that can happen during repair mechanism. So let's look at what people have used in the CRISPR-Cas systems in therapy alone. So I've shown already that Vertex is way ahead. They have their regulatory approvals for sickle cell anemia and beta thalassemia. So you can either do ex vivo, which is outside in the lab, or you do an in vivo genome editing. In an ex vivo, you take the cells out, you edit using CRISPR-Cas technology, and then after that you grow the cells, which are modified cells, and then transplant them back into humans. And this allows precise control and screening of the edited cells before you go for a patient infusion. It's widely used in clinical trials. This is the real gene therapy, but you do all your experiments outside, grow the cells, and then transplant. But what happens in vivo genome editing? You have all the CRISPR components, and they're delivered directly into the patient's body to the target tissues or cells. It could be the bone marrow or blood or so on. And this is all very important. You need to have it put in some LNP type of particles so that they don't get degraded before the delivery of these into cells or the blood system. There are fewer clinical trials due to ethical and safety concerns, but this requires highly efficient and targeted delivery methods. Therefore, people still prefer doing the CRISPR-Cas technology in the lab, growing the cells, and then re-infusion into the patient. Now, for delivery methods for in vivo, it could be choice of the delivery, it could be DNA, RNA, or RNP. It could be the method, could be electroporation or viral vectors or nanoparticles. And here, electroporation is common in ex vivo trials, and multiple strategies are being explored for effective in vivo delivery. So now let's look at what are the patent filings we are only looking at in therapeutic models or medical applications. Again, if you see, China stands out as compared to all other countries. We found about 640 odd applications. US had 583. They're really running neck to neck. And Europe had about 378. India, we looked at about 85 different applications. Australia has 205. So you can see here that China is standing out. Even though they never had any of the seminal patents, the seminal patents were mainly in the US between CVC and Broad. Of course, you had Toolgen from Korea, and then Sigma Aldrich and Villainous, but there are not too many of those. And therefore, it is the US, but China, the minute they understood the seminal patent importance, they took a license and then did applications into several medical technologies that they could do in therapy. And therefore, they are way ahead in patenting as compared to even Europe or India or Brazil or Australia or so and so. But most of the China ones are filed mainly in China, and they haven't gone so much to US or other countries, whereas US applicants are filed in US and China. Sorry. What are the various therapies that people have done? If you look at it, I'm not going back again because people have done it in base editing. They've done gene knockout or replacement. There are two technologies that they've used. But PAM is very important. I have to just tell you a little story about PAM. In Europe, CVC got the patent, they got enablement and everything. But these patents were opposed by several companies, including Broad. And one of the issues that CVC is facing now is that they did not talk about PAM in their seminal patent, because of which EPO has now come and said, look, you have an enablement problem because PAM sequences or PAM, the idea of PAM was not disclosed or enabled in the CBC pattern. And because PAM is very important, you should have enabled it. Though EPO said that the technology can be used from prokaryotes to eukaryotes, but one of the things that brought out is that look, you don't have PAM sequences, and if you don't have PAM sequences, this will not work. Without PAM, this will not work. And therefore, now CVC has a challenge where their patents, they want to give up their patents or may get revoked on enablement only for the point that PAM has not been disclosed or enabled in the EP patent of CVC, which is Jennifer Doudna and Emmanuel Charpentier. People have used a lot of this technology, and basically I said a lot of cancer, viral infections, liver diseases, cardiovascular diseases, ocular diseases. It could be brain or neurogenetic diseases, lung diseases, and metabolic. So when we looked at it, there's a whole lot of technologies that people have gone and used this CRISPR-Cas genome editing for treating human genetic disorders. As you see from the pie chart that I've shown, is that cancer, which is green in color, accounts for nearly 40% of all the applications. These are all patent applications that I'm talking about. We do have sickle cell anemia. We do have Duchenne muscular dystrophy, cystic fibrosis, HIV, and also cardiovascular pulmonary diseases. So main is actually cancer, sickle cell anemia, beta thalassemia, or most of the diseases where you can treat them through blood will be done, but people are also now looking at other solid tissues, cancer, and so on. The next part of my talk is on diagnostics. And so this is very transformative because even though it was all for gene editing, but they realized that CRISPR-Cas can also be used for detecting different diseases, such as cancer or infections, such as viral infection or bacterial infection. And people have used it across viruses, bacteria, chlamydia, tumor, parasite, and so on. One of this important thing is that people have generally used Cas12 and Cas13. And these are the different types of systems that people have used to show. Either it can be fluorescent signal or a lateral flow. But most important for you is most of the enzymes used are Cas12, Cas12a, or Cas13, 14, and so on. One of the important technologies from India is Feluda, again from CSIR, IGIB, where you have used an FN-Cas9, again a modified one, and they formed the FN-Cas9 RNP complex. I don't want to go through the whole thing, but very important is when you have this system and you put it into the strip, you can actually detect COVID. They use it for COVID and could identify whether you are infected or not. And so this was a very important thing from India, and this is called as Feluda, which is FN-Cas9 editor-linked uniform detection assay. Most of the findings, there are many of them, as I told you, there is Sherlock, there's Detector, there are many different systems. In my report, I've given all of that. But just to tell you that India is also in the race for FN-Cas9 and using that FN-Cas9 for detection, also using different Cas9 for therapy by CSIR and other institutes. If I look at diagnostic per se, 80% of the filings are in China, because the minute they identified this, they went and applied it to different bacteria, viruses, and so on. Another very important thing that you can see here is Cas12 was used by, I just want to tell you this, was used by Sherlock from the last two down. If you see there's Sherlock Holmes, they used Cas12, Cas13, and Holmes used this Cas12, and Broad Institute was responsible for use of detection of DNA or RNA using Cas12, and the Cas12a enzyme was by a license from China to Broad Institute, and this is the Tolo Biotech or the Shanghai Tolo. So China does have the rights for the enzyme Cas12 for use in diagnostics and detection. Ours in India is FN-Cas9. People have used Cas13. CVC is also, or University of California is also a main player for detection. And they have the many other things like detector and other for detection of COVID. They got quick approval, regulatory approval during COVID for use of CRISPR-Cas for detection of viruses. Again, China is leading for use of CRISPR-Cas for diagnostics. And they have about 425 applications or 445, whereas US is only 94. Other countries are much, much less. They've also used CRISPR-Cas in vaccines. And so you can either do a whole organism you can inactivate, or you can use it for subunit or nucleic acid. So people have used CRISPR-Cas for viral genome editing or the viral vector engineering. They've come up with novel platforms for precise insertion of immunogenic sequences, which can be then used as a vaccine. And they've also used host cell modification, where you can engineer cell lines for improved vaccine production and virus propagation. Again, if you see, China here has 30% of all the filings for vaccines. They've used it in various different types of vaccines, but they're mainly filed only in China. Only a couple of them have filed in other countries, whereas United States has filed in US as well as China. That's why the China number looks much bigger. So these are the two major geographies where you have the main applications getting filed. We do have some from Europe as well, but because there are so many countries, it looks less in number. But Europe is also a leader for the vaccines, use of CRISPR-Cas for vaccines. As I told you, it's another very important thing is people, because Cas9 was involved in a lot of litigation, people didn't know whom to license it and so on. And PAM was very important. You needed that PAM, which you see as NGG, which you have. And you also need this other sequences that have to be there for the Cas9 to work or the CRISPR-Cas technology to work. And therefore, people have come up with CRISPR-Cas with Cas12a enzyme, Cas13a, and Cas14. Feng Zhang has come up with Cas13 technologies. As I told you, Cas12 is with China, Tolo Technologies, and you also have Cas14, which is again used by some companies or some institutes. So these are the major enzymes. People have moved away a little from Cas9, but I must tell in therapy or therapeutic medical applications, it is still Cas9 or modified Cas9. There's dCas9, there are different types of Cas9, which we have covered in this variant. So variants of Cas9 are very important. You have dCas9, you have FN-Cas9, and modifications of Cas9 because you need strand-specific cleavage. And also very important is different Cas need different PAM, and therefore people are looking at different variations of Cas9, Cas12, and Cas13. Just to tell about FN-Cas9, again from CSIR IGIB, what is it? They've taken it from FN species. It is less pathogenic and it has enhanced variants for broader utility. And FN-Cas9 can target both positive and negative strand RNA. It can inhibit RNA viruses such as hepatitis C. It has a lot of advantages. You have dead Cas9, which is dCas9. It can repress or activate gene expression. It's good for epigenetic editing and so on. I would just like to quickly go there, using it for biological models. You can make different rodents, different cell lines, use CRISPR-Cas for editing. Again, you'll see China is way ahead, especially in different nuclear models, different cell models, animal models that China is doing. But they've generally filed all these applications mainly in China. And so if you look at other countries, we don't have too many where CRISPR-Cas patent applications for modifying a cell line or an animal. I didn't talk about it. China has also done yeah, he did produce twins where they modified the CCR5 gene and ran into a lot of ethical issues. So China is way, way ahead in CRISPR-Cas technology in a lot of medical applications, agriculture, fermentation technology, and so on. In India, again, who are the thing? People are working on biomedicine, agriculture, new Cas enzyme, using the applications in bacteria and fungi. Another important technology which I didn't talk about is CAR-T, where you are modifying the uh, genes or the cell lines using CAR-T for CAR-T technology, modifying the genes, etc using CRISPR-Cas. It's a very big thing where an intersection of CAR-T along with CRISPR-Cas. And there's several applications on that. Who are the important people in India who have filed is Shanghai Tolong, Broad Institute, CVC, Toolgen, Vertex, Villainous, Sigma, Aldrich, Editas, CSAR is there with FN-Cas9, CRISPR Bits has filed applications, TIGS has filed applications, NIPGR has filed applications. There are others, but I only took some of the major people in this technology which are from India. Thank you very much. I'm going to stop now. And any questions, I will be happy to answer.
Thank you, Malathi. That was, uh, as usual, a very informative talk. Can you hear me?
I can hear you.
Okay, good, good.
Mhm.
So while we'll ask for questions in the and there are some which are coming up. In the
Mhm.
meantime, I just wanted to. So there's a CRISPR case is actually I mean the case of CRISPR in patent law is there's a lot of stuff which to learn from, right? About
Yes.
drafting strategy, somebody missing out eukaryotes, right? Or
Mhm.
being first to file or being fast at filing and getting it granted, about enablement, about
Mm.
claiming priority rights in different jurisdiction and the rules being different, being early followers like the China story and so on and so forth. So it's
Mhm.
actually a very interesting case study.
Mhm.
Um, now, just a quick thing now with, say, some of the patents, say, being disallowed in Europe,
Mhm.
huh?
Yeah.
Does it open up any free space for latecomers, or there is a lot of other, there's a wall of other patents following that, which basically don't give any space for newcomers?
Just a minute. I just want to. I didn't give. There was a very nice um, um, one I wanted to share. Can I share that? There's
Yeah,
very nice thing. I didn't have time, but I would like to share something.
yeah.
Mm.
In the meantime, I have actually put down your report, right? Which
Yeah.
is available on the BFI
Yes.
website, right? I
Mm-hmm.
put it on the chat, okay?
Yes.
So that
Okay.
in case people want to read it, because I think it's
Sure.
definitely it's something which they should definitely read, right?
Yes. Yeah.
Um, okay, please go ahead. Yeah,
Yeah, so what we've done here is if you look at these are the five seminal patents or groups. So if you look at this is just Europe. So you were asking where is the seminal patents? Where are
yeah,
they? They're getting revoked in mainly Europe. US,
yeah,
it's very difficult to revoke the patents in US, PTAM. It's not that easy. It's very expensive. But in Europe, 9 months after the grant, you can go and oppose it. So this is University of Vilnius. If you look at it, They filed their patent application in March 2012. And the first patent application is revoked because of enablement issues. There are other issues. And there's one patent which is still pending. They're both from the same family. Okay. Don't worry about this opt-in and opt-out.
Yeah.
Then CVC, which I went only by date-wise. CVC,
Yeah.
if you know, the patent is finally revoked, one of the patents.
Hmm.
And they're going to give up one of their patents as well because of PAM. So
Yeah.
there is patent maintained in amended form, appeal is pending, patent application pending, granted opposition term pending, maintained in amended form. But there are their patents which are being revoked. Toolgen also, you'll see many of them opposition, and they are also being opposed by many people because they have that RNP. But if you look at it, it is 23/10/2012. Okay. The paper, they published the paper immediately after filing their application within a month or two. And so people were aware about the technology. But Toolgen is a very important South Korean company working, and they have some seminal patent. But in Europe, it seems to be really difficult because of the opposition procedure. But if you look at Sigma Aldrich, either they've withdrawn everything, they have revoked, and so on. But if I look at broad MIT, Harvard, and Rockefeller, they have some oppositions, but patents have been granted even after the opposition term, such as they maintained. So Rockefeller, along with Broad, there they have some patents, which are the seminal patents. But if I look at just Broad, MIT, and Harvard, because of the priority applications, they're all revoked. They're all revoked, and it is going to be quite tough for them to get this seminal patent, which is this 12 December 2012.
Yeah. Yeah,
Uh,
but these are all the European patents, right?
yeah, they're the European ones, correct.
Yeah. The US ones, I think they still
US are all active. Huh? All these are
yeah.
just Europe. Europe is also a big market. And uh, there is one more of Broad 25/7/2012. There is a granted patent and so on. I just wanted
Okay.
to highlight there that
Yeah, it's a very
in
interesting
Europe,
table.
yeah, it's
Is
a
it
very
from a
interesting
paper
paper.
or is it
There's a
from your
paper
report?
called Ulrich Storz. So if you put
Okay.
Storz, STORZ, he's a
Yeah.
patent attorney from Europe, and I took it from his paper.
Okay. So there was a question about Cas9, and I thought I'll take
Yes,
those up first. I
yes.
think you mentioned that for medical applications, Cas9 is going to be it's the main flagship, right? So
Not necessarily at present, yes,
okay,
because I'm going based on regulatory approvals, because that
okay,
was the main enzyme. You had variations of Cas9, and people, because they got approvals for Cas9, they're still continuing on that. But people
okay.
are moving away from Cas9, but those are the newer ones. But if you look at all the people who got the approvals, it's mainly on Cas9.
So Simanti Pal had a question, which is
Yeah.
basically that since Cas9 is already patented, will a modified version of the same protein, for example, FN-Cas9, also be granted a patent for an application, similar application?
It can be based on in first novelty, yes, because you've got a patent because it is different. But and you
Maybe,
also
maybe if I
no,
can
but
break it
one
up
I
a
want to
little.
make one correction here.
Yeah,
Cas9 enzyme per se, there is no patent on it.
exactly. Yeah, I was just going to
So I
come to
must
that.
tell people that. People always, though Cas9 is the important technology, CRISPR is not the technology that we use in a gene editing, correct? So we use the word CRISPR-Cas, correct? But if you look at all the technology, it's just guide RNA and Cas. So Cas9 per se, there is no patent on the enzyme because long back, people had identified Cas9, but they started modifying it for the PAM sequence. So you have D-Cas9, you have FN-Cas9, you have modified Cas9. There's a huge number of patents which are for Cas9. So you have to be careful when you say, oh, can I modify Cas9 and come up? Yes, you will definitely get a patent, but you have to be careful to see that you can get over the inventor step. You may get over the novelty, but whether you can get over the inventive step because Cas9 has been so well studied and there's so many papers on that. Okay, and that's why people moved away from Cas9 and Cas12, Cas13, Cas14 have all got very, very good applications.
Yeah, but will they also have FTO, freedom to operate,
For
FN
which
Cas9?
FN Cas9, I think so. They should have because see if you look at the main patent, it covers all Cas9. There's nothing like mutated Cas9, modified Cas9. So when you look at it, it's all Cas9 is covered. So when you read the patent, anyone with CRISPR-Cas using the guide RNA with Cas9, you're out. You cannot get a freedom to operate, and therefore that's why it is a little muddy there. And you may get an FN Cas9, you may get, but are you still infringing the initial patent? You may be, because Cas9 is very broad there. Use of
Uh,
Cas9 with guide RNA, okay? It's not just Cas9.
and have the seminal patents been filed in India?
Yes, some of them,
Yeah.
basically CVC, broad, toolgen, yeah. And
Okay.
there are also a lot of oppositions in India as well. Yes.
There's a
Maybe
question from
Sigma, Aldrich may not be,
yeah.
but all yes. CVC and broad, yes.
Okay. There's a question from Harvinder Kaur Kira on how does freedom to operate look like for Cas12a enzymes in India?
Cas12, they need to talk. It depends what you want to use it for. Cas12, I think right now is with Tolu, Shanghai.
12a, is it any different? 12a?
Yeah, 12A is what the main thing is for
Okay.
diagnostics. Mm.
Okay. Okay.
Mm. And
There's
we need to know
also
for what use it is, okay? So they don't have a patent for the enzyme per se. Remember that. It is
yeah,
for the use of the technology for diagnostics, okay?
diagnostics. Yeah.
Yeah. Detection, I
Uh,
would say, not even diagnostics, I'd say detection.
Yeah, there's a question from
Mm.
Kanhu Mishra asking one question many people have, including me, is if we want to do R&D using CRISPR-Cas9, how do we go about licensing?
So you will have to RNP, then you'll have to look at India patents. Toolgen may be one with RNP. There are others also with RNP. And we will have to look at what are the patents. First, we go and look at the patents which have been filed worldwide, and then come and see whether they have been filed in India, because it's a little difficult to just search our Indian Patent Office page. But if you know who are the main applicants, then based on the applicants, we can quickly search and look for those technology. So one I would like to say here, China earlier on, very early, I identified that CRISPR-Cas is a very important technology, and the government, or I don't know who it is, went and took a license for use in all medical technologies and everything. At that time, even the CVC or Broad didn't have an idea what is the value of that, and they got the license. But now, if you go and ask for a license, it's quite hefty and quite a lot. So China
Okay.
was way ahead in thinking out of the box.
Uh,
Yeah.
Dr. Prerna, before we proceed for the next question, just wanted to alert the audience. I've put the feedback link in the chat box. Please give your feedback because that helps us for curating good topics for you in the upcoming sessions. Yeah, over to you,
Yeah.
Dr. Prerna.
Yeah. I just also thought I'll just let people know that they can go back and look at the video. Um, Malti, I think there's a lot of content in the beginning, and there are people here who might have come from the patent landscape, but may not be as much from bio side of things. So for them, it will take a little while to digest
No, no, I think this
what you
is
said.
I know, no, I think this is very, very science intensive,
Yeah,
because without the science, I will not be able to go on the, what's a PAM, what's a
yeah.
DCAS9, and it's very highly technology focused, but also has insights on the patent side.
Okay. I have a few questions. When you
Yes, tell
showed
me.
those data and the graphs of geographical filings,
Yes.
The numbers you are showing, when you say China, it means filings in China or
Filings
owned by
in,
Chinese entity
no, filings in China.
in China.
And
So it
therefore,
could be
yeah,
filings by any other country also. So
correct.
it could be the Americans also, could be
Sure.
the Europeans also, and so on and so
Correct.
forth, right?
Yeah, or
So would
Koreans. Yeah.
yeah. So it probably, I mean, it does. Out of this, would you know what is the proportion which is by Chinese entities? Is it
Um,
majority of it?
yeah, so if I, let's say if I have 256 as a number,
Yeah.
uh, maybe about 70% of it would be Chinese. So
Okay.
when you see a big number in China, remember like vaccines or even medical technologies, they have gone and filed, especially I looked at diagnostics or vaccines, they've covered everything. I mean, there's nothing left.
Mm.
So this is where the numbers are high. Chinese have gone and filed everything in China, okay? And they've also gone filed some in Europe and some in US, a little in Europe, but some in US. But the US people have all filed in China. Not necessarily that they filed in Europe. They have filed in Europe, but all the US people have gone and filed in. Most of them have filed in China. So the China number looks high because Chinese applicants plus non-Chinese applicants are there.
Okay. You also mentioned this in incidents of where in the European courts, something which the CVC guys had filed was considered sort of something destroying inventive step for the Broad Institute's filings, but not so in the US,
Yeah,
right?
so
Uh,
yeah, that is
so
more on that's more on the priority. So Marifini's name was not in the final
No,
patent.
I didn't mean that. You had
Uh,
one more case where you said that, um, you know, the fact that for eukaryotes you could do it,
yes,
and not, and the fact that Broad had filed for eukaryotes could have been extrapolated from the filings of prokaryotes by the CVC group, right?
correct, correct,
That
correct.
was an issue also. So is
Yeah, but
that
yeah, so
the,
that's a very interesting thing. Let me take up Europe and Japan. So in Europe, when they looked at CVC patent, Broad and all said, no, 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 it's only should be with prokaryotes. Broad, we are the first for eukaryote. But they looked in EPO and said, the way it has been drafted, even though they have done experimental data with prokaryotes, it can be easily translated into eukaryotes. There's nothing great about the technology that I need to know, and a person skilled in the art can easily go and translate it into eukaryotes. And therefore, CVC got a very broad. So when you read it as CVC patent, it actually covers both prokaryotes and eukaryotes. It actually covers everything. And they said, no, 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 it covers both prokaryotes, eukaryotes, and everything. But however, the same patent is now maybe revoked in Europe because they did not talk about PAM. So same
yeah.
enablement issue, they said without PAM, it's not possible for the CAS to work. And therefore they said, no, no, it was very well known because PAM with Cas9 was well known before. And they said, no, no, it's quite well known. Why should I write about it? But they did write about it in their paper. So, the fight is like going into all these minute details, and to see that the seminal patterns are getting revoked in Europe is disheartening, but that's how they will fight the game.
Mm. Last question before we close. I know we've taken more of your time, but Datta Nadkarni had a question. What suggestions for the Indian Patent Office would you have based on your, uh, analysis?
Yeah, so as I told you, it took me long time, and I'm still learning every day about the CRISPR-Cas technology. And if you read the patent, it is quite difficult to understand the technology. I think for the patent office examiners, there is one patent where they say on section 59 they've revoked some patent, refused some patents, not understanding the technology, or they've given very broad patent for some of them, where Toolchain is now going and opposing those patents. You've got very narrow patents in Europe and US, but you've got a very broad patent in India. Or in some cases, they've given so many objections where most of these objections should not have been there. I think if I was in the place of the examiner or controller in India, it is difficult. It is difficult. For me, it took me 4, 5 months, 6 months to come to the level that I'm able to talk today. And you cannot expect an examiner to understand this technology, to allow it, or to refuse it. And therefore, our examiners and controllers need to keep themselves up to date on the technology. Similar with mRNA, similar with LNPs, there are challenges, and I think the challenges are more with the technology than with the examination.
Great. So I think on that note, we will close. Thank you
Yes.
once again, Malti. This is,
Mm.
I'm grateful for you to have presented this today. This report, I think, was supported by BFI Biome, right?
Yeah, correct.
Blockchain for Impact, and is now available for reading. So it helps. It's something which is a public resource to learn from. And you're forever the teacher, right? So thank you so much
No.
for
And I'm
doing
an
that.
ex-NCL, so I'm very happy that I did my PhD at NCL. I'd also like to say that if anybody wants to do some mRNA, I'm planning to do, but I need to get funds for it, for an
Mm,
mRNA landscape and LNP landscape, which is even tougher.
yeah,
That's something that I want to start on.
yeah. Great. I think those are both important topics. LNP also is very highly contested. So
Yes.
all the best. We'll also explore if something
Yeah.
is possible there. Thank you
Yeah.
so much. Thanks a lot. Over to Mugdha.
Yeah, thank you, Malti ma'am, for this session and alerting everyone again to once more. The feedback link is in the chat box. Please do give your feedback. I would like to announce the next session in this only one theme talk series, which is around patenting computer related inventions in India. And our speaker would be Sneha Kanetkar. This will happen on Tuesday, 7th of October at the same time, which is 4 o'clock. So do join us. Those who have already registered, you will get the alert once again. It is already there in your calendar. So please do join us at 4 o'clock on 7th of October. Thank you. Thank you all.
Thank you so much. Thanks a lot, Malti.
Yeah. Thank you.